Assalamualaikum class. Today we will talk about lipids. Biological lipids are a chemically diverse group of compounds, the common and defining feature of which is their insolubility in water. The biological functions of the lipids are as diverse as their chemistry. Fats and oils are the principal stored forms of energy in many organisms. Phospholipids and sterols are major structural elements of biological membranes. Other lipids, although present in relatively small quantities, play crucial roles as enzymes, cofactors, electron carriers, light absorbing pigment, hydrophobic anchors for proteins like chaperones to help membrane proteins fold, emulsifying agents in the digestive tract, hormone, and as intracellular messengers. This series of lectures is going to introduce you to representative lipids of each type with emphasis on their chemical structure and physical properties. We sometimes talk about fat as it were a very mean substance determined to destroy our look. But in fact, fat is a very important nutrient, and just like proteins and carbohydrates, our body needs some fats for energy, to absorb vitamins, and to protect your heart and brain health. For years, we have been told that eating fat is going to add inches to our waistline. It's going to raise our cholesterol level and cause a lot of health problems. But now we know that not all the fat is the same. In reality, fats are elegant little molecules. Each one is made up of three long hydrocarbon tails attached to a little hanger like molecule called cholesterol. This molecule is called a triglyceride. Triglycerides are the main constituent of body fat in humans and other vertebrates such as as well as vegetable fat. They are also present in the blood to enable the bidirectional transfer of adipose or fat tissues and blood glucose from the liver and are a major component of human skin oils. Like the other biological molecule, fats play essential role in the biology of humans and other organisms. Fats are just one type of lipid, a category of the molecules united by their inability to mix well with water. Lipids tend to be hydrophobic, nonpolar, and made up of mostly hydrocarbon chains, though there are some variations on this, which we will explore today. The different varieties of the lipids have different structures and correspondingly diverse roles in organisms. For instance, lipids store energy, provide insulation, make up cell membranes, form water repellent layers on leaves, and provide building blocks for hormones like testosterone. Here, we will look in greater detail at some of the most important types of lipids, including fats and oils, waxes, phospholipids, and steroids. Understand what a fatty acid is. A fatty acid is a carboxylic acid with a long aliphatic chain. Most naturally occurring fatty acids have an unbranched chain of an even number of carbon atoms ranging from 4 to 36 carbon. In some fatty acids, this chain is unbranched and fully saturated, which means that it doesn't contain any double bond. In others, the chain contains one or more double bonds and few contain three carbon rings, hydroxylic group, or methyl branches. In a fat molecule, the fatty acids are attached to each of the three carbon of the glycerol molecule with an ester bond through the oxygen atom. During the ester bond formation, three molecules are released. Since fats consist of three fatty acids and a glycerol, they are called triacylglycerols or triglycerides. Fats and oils, which may be saturated or unsaturated, can be unhealthy but also serve very important functions for plants and animals. Fats have important functions and many vitamins are fat soluble. Fats serve as a long term storage form of fatty acid and act as a source of energy. They are also providing our body with the insulation. At different types of fatty acids. Generally, fatty acids are classified in many ways. for example, by length, by saturation or unsaturation, or even as with the help of the carbon number, whether it is even or odd, and also by linear or branched nature. If we classify them according to their length, we have short chain fatty acids and fatty acids with aliphatic tails of 5 or less carbon. Then we have medium chain fatty acids. These are the fatty acids with aliphatic tails of 6 to 12 carbon, and they can form medium chain triglycerides. Then we have long chain fatty acids, and these are the fatty acids with aliphatic tails of 13 to 21 carbon. Very long chain fatty acids are the fatty acids with aliphatic tails of 22 or more carbon. Then we also have another category of fatty acid depending upon the number of carbon chains, if they are even or odd. Most fatty acids are even chained. For example, we have stearic acid, which has 16 carbon, and oleic acid, which has 18 carbons, meaning that an even number of carbon atom comprises them. Some fatty acids have odd numbers of carbon, and they are referred to as odd chain fatty acids. The most common odd chain fatty acids are the saturated C15 and C17 derivatives, and C here stands for number of carbon, and they are called pentadoconic acid and heptadoconic acid, and they are actually found in the dairy products. On a molecular level, odd chain fatty acids are biosynthesized and metabolized slightly differently from the even chain relatives. Beside the classification of fatty acid, depending upon their length or even or odd numbered chain of the fatty acids, we have this general classification of fats, whether they have a single bond or the hydrocarbon chain is linked with a double bond. So they are classified as saturated or unsaturated fat. I found a very nice video online, and it's pretty to understand the difference between the different types of saturated and unsaturated fatty acids according to that. So let's just watch that video.
What are the different types of fats? There are two general types, saturated fats and unsaturated fats. Unsaturated fats have three different subtypes, trans fats, monounsaturated fats, and polyunsaturated fats. To understand fats better, we need to start with the main form of fat, which is triglycerides. Triglycerides consist of a glycerol unit and three fatty acid chains. Fatty acids are made up of long chains of carbon atoms and hydrogen atoms. Some carbon atoms are linked by single bonds and others by double bonds. These bonds determine which type of fat the chain is classified as. The first type of fat we're going to look at is saturated fat. In saturated fat, all of the carbon atoms are saturated with hydrogen atoms and do not contain double bonds between the carbon atoms. This gives the molecule a linear formation. Saturated fats are usually solid at room temperature and have high melting points. Foods that are high in saturated fat include pork, fatty beef, cheese, whole milk, eggs, coconut and palm oils, and butter. The question is, are saturated fats healthy? Research suggests that saturated fat affects cholesterol levels by increasing overall LDL, increasing HDL, and increasing LDL particle size. You can learn more about cholesterol by checking out my cholesterol video, which is linked in the little I in the right-hand corner of your screen. The American Heart Association recommends limiting saturated fat intake to 5 to 6% of total daily caloric consumption. So according to that recommendation, if you're consuming 2,000 calories per day, no more than 120 calories should come from saturated fats. That's about 13 grams, which is 2 slices of cheddar cheese. I must note that there is a lot of research linking diets high in saturated fat to heart disease, and there is a lot of research showing that claim not to be true. As with any food, it's always about moderation. Now let's look at unsaturated fats. Unlike saturated fats, unsaturated fats have at least one double-bonded set of carbon atoms in their structure. This double bond can take on one of two formations. It can be a cis configuration or a trans configuration. In the cis formation, the hydrogen atoms are on the same side of the double-bonded carbon atoms, And in the trans formation, the hydrogen atoms are on opposite sides. Let's take a look at the trans configuration, or trans fatty acid. Trans fats are solid at room temperature and usually have a high melting point. There are natural and artificial trans fats. Natural trans fats, also known as ruminant trans fats, typically make up 2 to 5% of the fat in dairy products and 3 to 9% of the fat in beef and lamb. Several review studies have concluded that a moderate intake of ruminant trans fats does not appear to be harmful. Artificial trans fats are another story. Artificial trans fats are formed when manufacturers turn liquid oils into solid fats through a process called hydrogenation. Hydrogenation is a process by which vegetable oils are converted to solid fats simply by adding hydrogen atoms. Hydrogenation increases the shelf life and flavor stability of foods. Foods high in artificial trans fats include many fast food items, some baked items like cookies and cakes, frozen pizzas, some coffee creamers, fried foods, and stick margarines. So, again, the question is, are artificial trans fats healthy? Research suggests that diets in artificial trans fats affects cholesterol levels by increasing overall LDL and decreasing HDL. It is also linked to an increased risk of heart disease. Trans fats are believed to cause damage to the inner lining of blood vessels as well. In November 2013, the U.S. Food and Drug Administration made a preliminary determination that partially hydrogenated oils are no longer generally recognized as safe in human food. Many institutes recommend completely eliminating artificial trans fats from the diet. Keep in mind that products can be listed as 0 grams of trans fats if they contain 0 grams to less than 0.5 grams of trans fat per serving. You can also spot trans fats by reading ingredients lists and looking for the ingredients referred to as partially hydrogenated oils. The next unsaturated fat we're going to look at is monounsaturated fat. It has a cis molecular formation where the hydrogen atoms are on the same side. This gives it a bend or kink-like formation. Monounsaturated fats have only one carbon double bond in their molecule. They're usually liquid at room temperature and have lower melting points than saturated and trans fats. Foods that are high in monounsaturated fat include many plant-based oils such as olive oil, canola oil, and peanut oil. Other foods include avocados and many types of nuts and seeds. So, are monounsaturated fats healthy? They are thought of as generally being good for health, especially when chosen over saturated or trans fats. But it's always about moderation. Any fat can be unhealthy when consumed in unreasonably high quantities. The last type of fat on our list is polyunsaturated fat. It also has a cis molecular formation. Again, the hydrogen atoms are on the same side, also giving it a kinked formation. Polyunsaturated fats have more than one unsaturated carbon double bond in their molecule. They're typically liquid at room temperature, but start to turn solid when chilled. Polyunsaturated fats are generally classified by their omega numbering. The omega carbon is the carbon atom at the end of the hydrocarbon chain. There are 4 types of omega fatty acids: 3, 6, 7, and 9. These are determined by where the location of the first double bonded carbon atom is located. The fatty acid on the screen is an omega-3 fatty acid because the first double bond occurs at carbon number 3. The other omega fatty acids follow this same structure. The omega-3 and 6 fatty acids are considered essential because the body cannot make these. Foods high in omega-6 fatty acids include processed vegetable oils, salad dressings, fried foods, and grain-fed beef. Foods rich in omega-3 fatty acids include flax seeds, walnuts, salmon, and tuna. So, are polyunsaturated fats healthy? Research suggests that omega-6s can have inflammatory effects and omega-3s can have anti-inflammatory effects. So the ratio of these fats has been shown to be important. A ratio of 4 to 1, omega-6 to omega-3, or lower, has shown to decrease inflammation and lower the risk of other diseases. The average American diet currently has a ratio of 16 to 1. Once again, it's about moderation.
Look at some of the essential fatty acids. Essential fatty acids are the fatty acids required for biological processes, but they are not synthesized by human body. Consequently, they have to be supplemented through ingestion through the diet and are nutritionally very important. For example, we have omega-3 fatty acid or aliphatic alpha linoleic acid. They fall into this category, and this is one of the two fatty acids known to be very essential for human body. The other being omega-6 fatty acid or linoleic acid. These polyunsaturated fatty acids are called omega-3 because the third carbon from the end of the hydrocarbon chain is connected to its neighboring carbon by the help of a double bond. As I told you earlier, salmon, trout, and tuna are good sources of omega fatty acids. Research indicates that omega-3 fatty acids reduce the risk of sudden death from heart attack, reduce triglycerides in the blood, low blood pressure, and prevent thrombosis by inhibiting blood clotting. They also reduce inflammation and may help reduce the risk of some cancers in animals. In this figure, you can see um, different sources of omega fatty acids. For example, dry fruits, especially the nuts, and the fish such as salmon. Waxes are a type of long-chain non-polar lipid. Natural waxes are typically esters of fatty acid and long-chain alcohols. Waxes are synthesized by many animals and plants. Animal wax esters are typically derived from a variety of carboxylic acids and fatty alcohols. The composition of a wax depends not only on the species but also on the geographic location of the organism. The best known animal is, in the case of wax, is bee wax, but other insects also secrete waxes. A major component of bee wax is the ester merysyl palmitate, which bees use for constructing honeycombs. Spermaceti is also a wax that occurs in large amounts in the oil of a sperm whale's head. One of its main constituents is cetyl palmitate, which is an ester of fatty acid and fatty alcohol. Plant waxes are derived from the mixture of long-chain hydrocarbons containing functional groups such as an alkane, fatty acid, alcohol, diols, ketones, and aldehyde. Plants also use waxes as a protective coating to control evaporation and hydration and to prevent them from drying out. Waxes are valuable to both plants and animals because of their hydrophobic nature. This makes them water resistant, which prevent water from sticking on the surface. Unlike most natural waxes, which are esters, synthetic waxes consist of long chain hydrocarbons lacking functional group. Paraffin wax is a type of synthetic wax derived from the petroleum and it is refined by vacuum distillation. Synthetic waxes may also be obtained from polyethylene. Millions of these waxes are produced annually and they are used in adhesives, cosmetics, lubricants, insecticides, and in UV protection products. And they are, in fact, also found in the chewing gums. Now let's look at the phospholipids. Phospholipids are amphipathic molecules that make up the bilayer of the plasma membrane and keep the membrane fluid. Phospholipids are major components of the plasma membrane, which is the outermost layer of the animal cell. You all know that. And like fats, they are composed of fatty acid chains attached to a glycerol backbone. Unlike triglycerides, which have three fatty acids, phospholipids have two fatty acids that help them to form a diglycerol. The third carbon of the glycerol backbone is also occupied by a modified phosphate group. Look at the structure of a single phospholipid on the right side and you can see all those groups that I have just told you about. Now, however, just a phosphate group is attached to a diacylglycerol, it does not qualify to be a phospholipid. This would be considered a phosphatidate or the precursor of a phospholipid. To qualify as a phospholipid, the phosphate group should be modified by an alcohol. And we have some example. For example, we have phosphatidylcholine, which is an important phospholipid that are found in the plasma membrane. A phospholipid is an amphipathic molecule, as I told you, which means that it has both a hydrophobic and a hydrophilic component. A single phospholipid molecule has a phosphate group at one of the end, and that's called its head. You see this blue part in the right side of the figure? That's the head, and the two sides of the fat chains of the fatty acid that make up the lipid, they are the tails, which are like the white part in the phospholipid molecule, which is shown on the right side of this figure. Now, the phosphate group is negatively charged and it makes the polar head and hydrophilic region, or it is also called the water loving end of the phospholipid. And the phosphate heads are thus attracted to the water molecule in aqueous environment. The lipid tails, on the other hand, they are uncharged, nonpolar, and hydrophobic in nature. Therefore, they are called water-fearing. A hydrophobic molecule repels and it is repelled by the water. Now, some lipid tails consist of a saturated fatty acids and some consist of unsaturated fatty acids. This combination adds to the fluidity of the tails and they are therefore constantly in motion. Now in the figure, you can see a phospholipid molecule with two fatty acids and a modified phosphate group which is attached to the glycerol backbone. The phosphate may be modified by the addition of charge group or polar chemical groups. Two chemical groups that may modify the phosphate, for example, choline or serine, they are shown here. And both choline and serine attach to the phosphate group at the position which is labeled as R, and it can be like anything. For example, it can be a nitrogenous chemical group, and they are like labeled, they are like linked with the help of hydroxyl group, and they are like indicated here. Now, when we look at the phospholipids of the biological membrane, you know that cell membrane consists of two adjacent layers of phospholipids which form a bilayer. The fatty acid tails of the phospholipid face inside, away from the water, whereas the phosphate head face the outward aqueous side. Since the head face outward, one layer is exposed to the interior of the cell and one layer is exposed to the exterior. As the phosphate groups are polar and hydrophilic, they are attracted by water in the interstellar fluid. And because of the phospholipid chemical and its physical characteristics, the lipid bilayer acts as a semi-permeable membrane. And you know that when a membrane is semi-permeable, only lipophilic solutes can easily pass through this phospholipid bilayer. And as a result, there are two distinct aqueous compartments on each side of the membrane. This separation is essential for many biological functions, including cell communication and metabolism. Membrane fluidity of the plasma membrane. It is due to, for example, is because of many different type of lipids. For example, we have cholesterol within the phospholipid bilayer. Biological membrane remain fluid because of the unsaturated hydrophobic tails, which prevent phospholipid molecule from packing together and forming a solid molecule. If a drop of phospholipid is placed in water, like you see on the right side, the drop which is like it has white yeah, polar heads and the yellow tails, the phospholipid is going to like spontaneously form a structure that is called a micelle. So the structure that you see on the right side of the slide is a missile, and it has its hydrophilic heads, which are oriented outward to the water. And a missile are lipid molecules that are going to like arrange themselves in a spherical form in aqueous solution, exactly like the way it is shown here. Now, the formation of a missile is a response to the amphipathic nature of the fatty acid, meaning that they contain both hydrophilic and hydrophobic regions. So now we have steroids and sterols. Now, unlike phospholipids and fats, steroids have a fused ring structure like the one that you can see on the right side. Inside the circle, you see the rings which are fused together. Although they do not resemble the other lipid, they are grouped with them because they are also hydrophobic and insoluble in water. Now, all the steroids have four linked carbon rings, and many of them, like cholesterol, you can see the formula, they have a short tail. The steroids also have the hydroxyl functional group, and these steroids are classified as alcohol or sterols. Cholesterol, which is the important sterol, it is not only important, it's also the most common steroid, and it is mainly synthesized in the liver. It is the precursor of vitamin D, and cholesterol is also a precursor of many other biologically important steroid hormones like estrogen, testosterone, progesterone, which are secreted by the gonads and the endocrine glands. Therefore, steroids play a very important role in the body's reproductive system. Cholesterol also plays a role in synthesizing the steroid hormone aldosterone, which is used for osmoregulation, and also cortisol, which plays an important role in metabolism. Cholesterol is also the precursor of bile salts, which help in the emulsification of the fat and their absorption by the cell. It is also a component of the plasma membrane of the animal cells and the phospholipid bilayer. And as I told you, it was also playing a major role in membrane fluidity. Now, being the outermost structure in the animal cell, the plasma membrane is responsible for the transport of material and cellular recognition, and it is also involved in cell to cell communication. Thus, steroids also play an important role in the structure and function of the membrane, and it has also been discovered that steroids can be active in the brain where they can affect the nervous system. These steroids are called as the neurosteroids, and they are able to change the electrical activity in the brain, and they can either activate or tone down receptors that communicate messages from neurotransmitters. Since these neurosteroids can tone down receptors and decrease brain activity, steroids are also used in anesthetic medicines. Here you can see the structure of cholesterol and the cortisol and the fused ring structure, which is common among all the steroids and sterols. Let's look at the plasma transport and regulation and absorption of cholesterol. As an isolated molecule, cholesterol is only minimally soluble in water. Because of this, it dissolves in the blood at exceedingly small concentration. To be transported effectively, cholesterol is instead packaged within lipoproteins, complex discoidal particles with exterior amphiphilic proteins and lipids. Their outward facing surfaces are water soluble and their inward facing surfaces are lipid soluble. This allows it to travel through the blood through emulsification. Unbound cholesterol, being amphipathic, is transported in the monolayer surface of the lipoprotein particles along with the phospholipids and proteins. Cholesterol esters bound to the fatty acid, on the other hand, are transported within the fatty hydrophilic core of the lipoprotein along with the triglycerides. There are several types of lipoproteins in the blood. In order of increasing density, there are chylomicrons, very low density lipoproteins, intermediate density lipoprotein, low density lipoprotein, and high density lipoprotein. Lower protein to lipid ratio makes for less dense lipoproteins. Cholesterol within different lipoprotein is identical, although some is carried as a lipoprotein in its native form or free form, which is free from the alcohol, while other are as fatty acyl esters, which are also known as cholesterol esters within these particles. Now, first of all, let's discuss the LDL or the low density lipoproteins. Low density lipoproteins or LDL particles are the major blood cholesterol carriers. Each one contains approximately 1,500 molecules of cholesterol ester. Both LDL and its receptors form vesicles within the cells by the help of endocytosis. These particles then fuse with a lysosome, where the lysosomal acid lipase enzyme hydrolyzes the cholesterol ester. The cholesterol then can be used for membrane biosynthesis or esterification, and it is stored within the cell so as not to interfere with the cell membrane. Low density lipoprotein receptors are used up during cholesterol absorption, and the same protein that controls the synthesis of cholesterol de novo according to its presence inside the cell. A cell which is with abundant cholesterol will have its LDL receptors synthesis blocked to prevent new cholesterol in LDL molecule from being taken up. Conversely, LDL receptors synthesis proceeds within a cell is if a cell is like deficient in cholesterol. When the process becomes unregulated, low density lipoprotein molecules without receptor begin to appear in the blood. These low density lipoprotein molecules are then oxidized and then taken up by the macrophages, which become engorged and form foam cells. These foam cells often become trapped in the walls of the blood vessels and result to the formation of a plaque. That plaque is called as atherosclerotic plaque. Difference in cholesterol homeostasis affects the development of early plaque formation. These plaque are the main cause of heart attacks, strokes, and other serious medical problems, leading to the association of so-called LDL cholesterol, actually a lipoprotein, with bad cholesterol. High density lipoprotein particles are thought to transport cholesterol back to the liver either for excretion or for other tissues that synthesize hormones in a process that is known as reverse cholesterol transport. Large number of high density lipoproteins correlate with a better health outcome, whereas low number of high density lipoprotein is associated with many diseases of the arteries and progression in the arteries. So let's just sum up what we have learned so far about lipids. So, first of all, lipids are water insoluble cellular components of diverse structure that can be extracted by like non-polar solvents. Almost all the fatty acids, which are the hydrocarbon compounds of many lipids, have an even number of carbon atoms, usually 12 to 24, and they are either saturated or unsaturated with the double bonds almost always in the cis configuration. Triglycerols contain three fatty acid molecules esterified to the three hydrocarbons of the glycerol. Some triglycerols contain only one type of fatty acid, which are mixed with like triglycerols or two or three types. The triglycerols are primarily storage fats and they are present in many foods. The polar lipids with polar head and non-polar tails are major components of the membrane. The most abundant are the glycerophospholipids, which contain fatty acid esterified to two of the hydrocarbon groups of the glycerol and a second alcohol, the head group esterified to the third hydroxyl of the glycerol with the help of a phosphodiester bond. Other polar lipids are the sterols, and sterols have fused ring structure and the hydroxyl group. Cholesterol, which is a major sterol in the animal, is both a structural component of the membrane and a precursor to a wide variety of steroids.